Welcome back to NPTEL online certification course on novel drug delivery systems. I am Dr. Satish Dyavanapelli, Assistant Professor at Department of Pharmaceutical Sciences and Technology, Institute of Chemical Technology, Mumbai. In novel drug delivery systems, we started with the controlled drug delivery systems. In our previous class, we have discussed on the introduction to controlled drug delivery systems. In previous class, we have discussed what are the controlled drug delivery systems, and then we have seen what is the need of controlled drug delivery systems in a pharmaceutical industry. And further, we have focused what are the rationale for developing the controlled drug delivery systems. And after that, we have seen what are the advantages in terms of reducing the dose and the dosing frequency and improving the patient compliance and also patient convenience. And then we have seen some of the limitations, which are the nothing but the dose dumping and the high cost and some other parameters we have discussed. And later, we have seen what are the challenges which are involved in designing and development of the controlled release drug delivery systems. Today, we are going to see the fundamentals on the approaches to design controlled release drug delivery systems. So, here we are going to see the knowledge of the approaches which will involve in the designing of the controlled release drug delivery systems. So, these are the today's my flow of presentation. So, first I will discuss what are the general design considerations of controlled release drug delivery systems. Further, I will focus on the key factors which are important in the designing of the controlled release drug delivery systems, and then we will see the strategies to design controlled release drug delivery systems. But in our upcoming classes, we are going to focus on this. Today, we are going to see only the overview of the strategies, and then we will end up our presentation with the characteristics that make a drug unsuitable for controlled release drug delivery systems, that which we not seen in previous presentation, and today we are going to discuss on these parameters. So, general design consideration of controlled release drug delivery systems. So, designing a controlled drug delivery systems that must be balance two parameters, that is the formulation related factors and then the drug related factors. So, here while designing of any controlled release formulations, we need to consider these drug related as well as formulation related parameters, which involves the materials and the stability of the formulations and the pharmacokinetics with the drug characteristic like protein binding and the permeability parameters. So, we need to balance these two things while designing the controlled drug delivery systems. All this must align with the regulatory standards for safe and effective therapeutic outcome. When you maintain quality, remember these three words. When you maintain the quality, safety and the efficacy, from the designing to the development, the chances of getting approval or compliance is very, very high in the regulatory process, because during the selection of your raw material and the designing of your formulation, your quality need to be included during the design step. And once you input or once you consider quality as a parameter from the selection to the designing and then for the development, and the chances of the getting high quality is very, very high. So, the maintaining quality, we need to balance quality as well as quantity. At the same time, you need to focus on safety and efficacy. So, if you maintain these three parameters, quality, safety and efficacy as well as quantity, that is important for scalable. So, then the chances of regulatory is very, very high compared with the low standard and the low quality, non-effective and toxic products. So, we need to consider these three parameters. So, definitely that will depends on your drug as well as the formulations. So, while designing, we need to consider these are the two general considerations. So, here we are going to see what are the properties or parameters. So, here first we can classify the general design considerations, formulation related parameters, and second one is drug related parameters. So, here you can see the biomaterial properties, and here you can see the list of the what are the biomaterial properties. So, which includes biocompatibility of the materials and the hydrophilicity, surface chemistry, the degradation, rheological and mechanical properties of your materials. So, that is very, very important. Based on that, we can design a right and the best control drug delivery systems. And yes, not only designing of that and considering biometric properties, we need to consider the route of administration. So, after designing of a right and best control formulation, but if you do not give through right route, and your dosage form will not work out and will not show the control properties. So, we need to consider what is the route of delivery. So, we have oral, topical, ocular and parenteral and other types of the delivery systems, other types of delivery routes, that is nasal and other systems. So, we need to focus on what is the delivery route, and then the pharmacokinetics parameters also we need to consider. So, that we are going to see in today's presentation, therapeutic index and therapeutic window, drug release mechanism and the dosage forms. And also your formulation need to stabilized, because here we are going to use various biological molecules and that need to be stabilized. And when you are taking any nano systems, that is need to be maintained. And whereas in case of here, we can also focus on the active and targeting and passive targeting mechanism. So, we need to consider while designing the control drug delivery systems. And yes, these are the biological barriers, so that need to be overcome by using the control drug delivery systems. So, while designing, we need to see which route we are giving and what are the barriers. For example, in case of brain delivery, we know that blood brain barrier is one of the major barrier which will control the permeation of drug to the brain. So, we can try with the oral routes, we can try with the nasal routes and other brain targeting systems. So, with that we can improve the drug profile in the brain. So, we need to consider what are the brain barriers, what are the biological barriers. So, here we need to focus on the regulatory aspects, the animal models and the clinical studies during the design, and also the historic evaluation whether the formulations are available on the macro and the micro size and the nano size. With these, so in general consideration, we can look on the formulation related parameters and the drug related parameters. Both we need to important, because these drug related when you know, when you have the knowledge on the drug related parameters, we can easily make a best or better formulation by using these general design considerations. So, here now I am going to discuss on some of the key factors which are involved in the designing of the controlled release drug delivery systems. So, as we know, this can be classified into three things. First, we are going to see on the biopharmaceutical properties. So, here we can see these are the various biopharmaceutical parameters, and these are the desired features which are required for a candidate to design as a controlled release delivery formulation. So, as we know that molecular size is very important. So, when you have a low molecular weight, the absorption will be very fast and it will undergo the complete absorption when you have the low molecular size. So, preferably 300 to 400 Daltons, and yes, you can go ahead with the less than 600 Daltons. And solubility is very important parameter in case of pre-formulation. So, your drug must be shown solubility the more than 0.1 mg per ml. So, once your drug is solubilized, that can during the dissolution or in the body. So, if your drug is solubilized, then it can convert into the desired physiological mediums or in the dissolution medium. So, for that the minimum is the 0.1 mg ml is very important for as a desired feature for the designing of the controlled release drug delivery systems. So, if your drug is water insoluble and we need to convert or we need to use various solubility enhancement techniques like solid dispersion and complexation, or by using hot melt excretion, otherwise converting of crystalline drugs into the amorphous forms and co-solvency, various techniques we can improve the solubility, and then we can use such modified drugs and we can convert into controlled release drug delivery systems. Otherwise, your drug is not aqueous soluble, then we cannot expect the release from the dosage form. Now, as we know the partition coefficient, so the concentration of the drug in the oil phase by the concentration of the drug in the water phase, that should be in the 1 to 2 range, which is very important for the permeability. So, after release the drug will come out from the system. So, such cases we need to maintain the optimum partition coefficient, and then the another parameter is dissociation constant. So, this is very important, because for a drug when it required to permeate through various biological barriers, your drug need to be in the unionized form. So, to get into the unionized form, so the dissociation constant is very important. Acid drugs, as we know, which will not dissociate are in the acidic medium. Similarly, basic drugs will not dissociate in the basic medium. So, such cases we need to consider if your drug is acidic drugs and that pK val should be more than 2.5, whereas if your drug is basic drugs, your dissociation constant should be less than 11. So, that is very important for the permeation. And then ionization at physiological pH, that should be very important. Your ionization at physiological pH should be less than 95%. Then stability in the GI media, and that should be stable in both gastric and intestinal pH. If your drug is not stable, we need to design various strategies like entry-coated formulations. With that, we can avoid the acidic degradation. So, if your drug is stable in both gastric and intestinal pH, so you no need to convert the drugs into the desired format. So, here the absorption mechanism is also very important. As we know, the most of the drugs will follow the passive mechanism and but not through a window. If the drugs follows any the absorption window, then again, so we need to modify your strategies. So, these are the some biopharmaceutic properties which are important for the control release drug delivery systems. Here we are going to discuss on the various pharmacokinetic parameters for a drugs. So, we need to consider not only biopharmaceutical properties, we need to consider pharmacokinetic properties while designing the control release drug delivery systems. Now, we know that after administration, drug will follow various pharmacokinetic properties or you can say pharmacokinetic profiles. So, here it comes the absorption and then distribution, metabolism and excretion, which also known as ADME. Pharmacokinetics is nothing but what body does to the drug after administration, how body will take the molecule or take the dosage form from various parameters like absorption, distribution, metabolism and excretion. So, this is called as pharmacokinetics. So, yes, in the absorption, right, site included in the mouth, stomach, small bowel and their various places the drug will absorb. So, absorption is nothing but it will crosses the various biological barriers and it will reaches to the blood, that is called as the process is called as absorption. And there are so many limitations are there and it will depend on the your drug properties. And then, so drug will enter into the blood and then it will distribute throughout the body, which is called as distribution. So, here we have lot of compartments, so which is nothing but plasma and extracellular, intracellular spaces and other compartments. Further, after complete distribution and then after certain time intervals, drug will undergo the metabolism and you will get the various drug metabolites and of course, then it will follow the excretion, then drug can be excreted. So, all these parameters we need to consider. So, here in case of pharmacokinetic parameters, we need to consider absorption rate constant, that is very important. Your absorption rate constant should be very high for a control release drug delivery system. And elimination half life, that should be 2 to 6 hours. It should not be less than and it should not be more than of the optimal elimination half life. And metabolism rate, that should not too high, otherwise your drug will not survive in the blood and it will metabolize immediately and that should be controlled. And then dosage form index peaks, here it maximum concentration of the drug upon the minimum concentration of drug in the plasma. So, that ratio should be 1. So, this dosage form index also we need to consider. So, maximum concentration of the drug in the plasma versus maximum concentration of drug in the plasma by the minimum concentration in the plasma. So, that gives the dosage form index. So, while designing any dosage, we need to consider dosage form index that should be below 1. So, here we are going to discuss on the pharmacodynamic parameters. So, these are the desired features. So, yes, in a control release form, your maximum dose is 1 gram. So, we should not go more than 1 gram, otherwise very difficult to design and will face lot of problem during the formulation and packaging also. So, that need to be your dose should be the 1 gram. And here we will see some of the important parameters which are very important, that is the therapeutic window and the therapeutic index, that should be very wide for a control release drug delivery systems. So, as we know the definition here I have mentioned for understanding, Therapeutic window is the concentration range of the drug that balances adverse and therapeutic effects, right. That should be wider, maximum. Now, we can see with this graph, here you can see the exposure and here is the percentage of patient responding, and you can see the green line speaks about the efficacy, and here you can see the ED50 value and the TD50 value, that is toxic dose. And you can see the therapeutic window with this the green line. So, that should be maximum. So, when you have a high therapeutic window, you can easily select, you can go for the control drug delivery system. And we will see what is the therapeutic index. Therapeutic index is defined as the dose required to produce in the 50% of the population, that is ED50, divided by the dose required to produce the toxic effect in 50% of the population. ED50 by TD50, that is nothing but the effective, that is nothing but the dose required to produce a response in 50% of the population, and the toxic effect and the response, or you can say the efficacy in 50% of population. So, that will give the therapeutic index. So, this both should be higher, and the relationship between the pharmacokinetic and pharmacodynamic, that should be good. That means, the ADME properties will help in the dynamic or in the efficacy of the drug. That relationship should be very, very good. So, then we can go for the control release formulations. If you would like to know more, here is the reference. You can go through here and you will understand various the important parameters of pharmacokinetics. And here we are going to see some of the strategies which will help in the designing of the control release drug delivery systems. So, here we can classify this into various strategies. The first one is the chemical approach, and then biological approach, and third one is the pharmaceutical approach. So, the drugs, the release mechanism will depend on the chemical approach, that either by the pH and by using the ion and by the hydrolysis. So, these are the three strategies we will see in the chemical approach. The materials which will stimuli or which will undergo the responsive as in a pH, one of the mechanism, either by ion or by the hydrolysis activated. But while designing of these systems, your drug properties should not be modified by using of these materials. So, during the development, we need to see the compatibility of your materials while going with the chemical approach. That should not affect your therapeutic efficacy as well as safety and in the final degradation product. So, these three things we need to consider while designing. We need to check the compatibility of the control release material which are undergoing to sensitize at these conditions, and then your active molecule, and that should not affect physical properties as well as chemical and the biological response of your drug molecule. So, while designing we need to consider, so we need to do as many as studies, comparative studies with your materials as well as with your active drug. So, then we can go based on the various chemical approaches and pH activated, whereas when you want to load the sensitive molecules which are undergo in acidic conditions by using entry coated polymers. So, these drugs will sensitize in the entry coated, and if you are converting it a control release formulation, and the drugs will release only in the intestinal pH. So, similarly based on the various physiological conditions we can target our formulation at various locations. And the second one is biological approach. Here we can focus on the enzyme activated and the biochemical activated. So, by having of the stimuli with the various enzymes and the biochemical reactions, we can select the right material and we need to design the biological approaches as in a control release drug delivery systems. And the third one is pharmaceutical. Third one is the pharmaceutical approach. So, here what we do, we will control the release. Here we will control the release by taking dissolution as a key parameter, and the second one is the diffusion. The pharmaceutical approach can be classified into various strategies, in that the dissolution control release mechanisms and then diffusion control release mechanism and the combinations, both dissolution and the diffusion control systems. So, this further dissolution can be classified into the matrix dissolution control systems and then encapsulation or reservoir dissolution control systems, whereas diffusion can be classified into reservoir devices and the matrix devices. So, all these strategies we are going to learn in our upcoming classes. So, here is the just overview of these strategies. So, and there are some characteristics that make a drug unsuitable for control release drug delivery systems. If your drug follow any of these characteristics, so which cannot be suitable for control release drug delivery systems. Yes, but after modifying with their properties, then we can utilize it for the control release drug delivery systems. So, here short elimination half life, it may happen through various issues that need to be overcome. If a drug have a short elimination half life, if you are giving control release formulation, your drug will release and it will reach into the blood, but if a drug is very short half life, there is no use of converting into control release drug delivery systems. And as we know, we discussed therapy index should be wider, but if it is very short, then we cannot use it for the control release drug delivery systems. And if a drug is poor absorption, we cannot use it for the control release drug delivery systems. We can use some strategies by improving the permeability, by converting or conjugating with various lipid molecules, or by using lipid delivery systems, then we can use it for the control release delivery systems. And if a drug has high plasma protein binding, it will show the high half life, and such case also it will not possible, your drug will not distribute, and that will not fit for the control release drug delivery systems. So, the plasma protein binding should be optimum for your drug. Such drugs can be, we can use it for the control release drug. Extensive first pass metabolism also is not suitable. So, we need to convert that drugs or the dosage forms into another route of delivery, then we can go ahead with the control release drug delivery systems. And the slow absorption is also limitation of such drugs. So, that can also be overcome by various strategies for the enhancement of the absorption. So, these are some of the characteristics of the drugs which are not suitable for the control release drug delivery systems. So, with this, today I am going to stop today's lecture. So far we have discussed what are the approaches to design as in a control release drug delivery systems. Here we discussed general considerations, and then we have seen the key factors, and then we have seen the various characteristics and some are the strategies which are involved in the designing of control release drug delivery systems. So, I am going to stop here. Thank you so much. In our upcoming class, we are going to discuss other strategies which we can use it and utilize it for the designing of the drug delivery systems. Thank you so much.